Here, let's going to focus on the topic of gluconeogenesis. And now let's begin with the simple definition of gluconeogenesis, and which is a process where our body makes new glucose from a non-carbohydrate sources. The main process of gluconeogenesis are lactate that are produced from the muscle during intense exercise. Gluconeogenesis primarily takes place in the liver and to the lesser extent in the kidney, and the condition for the gluconeogenesis are fasting or starvation. Gluconeogenesis occurs to maintain the blood glucose level when the dietary glucose is unavailable, and this condition happens during our prolonged fasting or starvation. And in the intense exercise, the body produces glucose to meet energy demand when muscle glucose stores are depleted. And also in the hormonal signals, hormones like glucagon triggers the gluconeogenesis to raise blood glucose level during stress or low glucose conditions. Gluconeogenesis helps to prevent the hypoglycemia, which is the conditions where blood glucose level drops too low. Now let's focus on the substrates of gluconeogenesis. During intense exercise, muscle produces lactate from glucose through glycolysis when the oxygen is limited. Lactate is transported from muscles to the liver through bloodstream. In the liver, lactate is converted back into the glucose through gluconeogenesis, and now the amino acid, especially alanine, released from the protein when the body breaks down them. Alanine is transported from the muscles to the liver through bloodstream. In the liver, alanine is converted into the pyruvate through a process is called as transamination. This process involves the transfer of an amino group from alanine to a molecule called alpha ketoglutarate, resulting in a pyruvate and glutamate. Then glycerol comes from the breakdown of fats, that is triglycerides, in the fat tissues. Here, the glycerol, with the help of an ATP and glycerol kinase enzyme, it transforms into glycerol 3-phosphate, and this transforms into dihydroxyacetone phosphate. With the help of an enzyme, glycerol 3-phosphate dehydrogenase, and this dihydroxyacetone phosphate enters into the pathway of either glycolysis or gluconeogenesis based on the body condition. And hence, now the pyruvate can be formed from lactate, alanine, and also from the glycerol, and then the pyruvate is converted into phosphoenol pyruvate. Now let's discuss about the steps of gluconeogenesis in detail.
Pathway of gluconeogenesis. First, the pyruvate in the cytoplasm enters into mitochondria. Then pyruvate is converted into oxaloacetate by the enzyme pyruvate carboxylase. The oxaloacetate must be transported from mitochondria to cytosol because the further reactions of gluconeogenesis takes place in cytosol. This is achieved by the malate aspartate shuttle. Oxaloacetate is first converted into malate by the enzyme malate dehydrogenase in mitochondria. Then the malate is transported to cytosol through malate aspartate shuttle. In cytosol, malate is reconverted to oxaloacetate by the enzyme malate dehydrogenase. Malate dehydrogenase enzyme is present in both mitochondria and cytoplasm. In cytoplasm, phosphoenol pyruvate carboxykinase converts oxaloacetate to phosphoenol pyruvate. This helps to reverse the irreversible reaction, that is the step 9 of glycolysis, mediated by pyruvate kinase. Then, phosphoenol pyruvate undergoes further reaction. The freely reversible reactions of glycolysis, step 8, 7, 6, 5 and 4 gets reversed. Now, fructose 1,6-bisphosphate is converted to fructose 6-phosphate by the enzyme fructose 1,6-bisphosphatase. Fructose 6-phosphate is isomerized to glucose 6-phosphate by the freely reversible reaction catalyzed by hexose phosphate isomerase enzyme. Then the first step of glycolysis, that is glucose 6-phosphate, is converted to glucose by the enzyme glucose 6-phosphatase. The glucose 6-phosphatase enzyme is present only in the smooth endoplasmic reticulum. The glucose 6-phosphate produced in the cytosol is transferred to smooth endoplasmic reticulum through the transporter T1. The glucose 6-phosphatase converts the glucose 6-phosphate to glucose. This glucose is transported out of the smooth endoplasmic reticulum to enter into the cytosol by the transporter T2. And the glucose in the cytosol is released to the bloodstream by the hepatic vein.
Energetics of gluconeogenesis. Gluconeogenesis is an energy consuming process. Pyruvate carboxylase, PEP carboxykinase, phosphoglycerate kinase, it all requires 1 ATP each. Pyruvate is converted into half glucose by 3 ATP. 3 ATP are utilized to produce half glucose, or 6 ATPs are utilized for the production of 1 glucose.
Let's see about the molecules involved in the gluconeogenesis and their fates. So first one is the fate of pyruvate. Pyruvate is the end product of glycolysis, which is the breakdown of glucose, and is also converted into glycerol. And then in anaerobic condition it is converted into lactate, and in amino acids it is a precursor of so many compounds such as alanine, aspartate, glycine, serine, and cysteine. Next is aryl acetate, which is converted into phosphoenol pyruvate, then glucose 6-phosphate, then glucose, then acetyl CoA, which is a major component which runs the TCA cycle for the ATP production. Fates of oxaloacetate. Oxaloacetate is converted into pyruvate, and it also takes part in the participation of TCA cycle. In amino acid synthesis, it is converted into aspartate, through aspartate, oxaloacetate, then PEP, then malate, in malate aspartate shuttle. Then fates of glucose 6-phosphate. Glucose 6-phosphate takes part in the glycolysis, where pyruvate, it is converted into pyruvate, then TCA cycle, then NEET cycle, it goes on. Next is gluconeogenesis, where fructose 6-phosphate is converted into glucose 6-phosphate through the enzyme phosphoexokinase. Next, gluconeogenesis, which is converted, glucose 6-phosphate is converted to glucose 1-phosphate through the enzyme phosphoglucomutase. Then a metabolic pathway known as the pentose phosphate pathway, where glucose 6-phosphate plays a major role in this pathway. So as we know, gluconeogenesis is a very vital metabolic process, and we generate glucose from non-carbohydrated sources also, like amino acids, lactate, and glycerol. So it has so many clinical significance. Let's see 4 of it. First is hypoglycemic prevention, that is, gluconeogenesis prevents hypoglycemia. Hypoglycemia is a condition where we have low blood sugar by generating glucose from alternative sources. Next is type 2 diabetes. Dysregulation of gluconeogenesis contributes to insulin resistance and hyperglycemia. Hyperglycemia is a condition where your blood sugar level is high, increasing the risk of developing type 2 diabetes. Next is metabolic syndrome. Gluconeogenesis is often upregulated in metabolic syndrome, exacerbating insulin resistance and hyperglycemia. Next is ketoacidosis. In diabetic ketoacidosis, excessive gluconeogenesis can lead to life-threatening ketoacidosis.
Role of ATP. Phosphorylation reaction. Overcoming energy barriers. Many reactions are energetically unfavorable under certain conditions. ATP hydrolysis is provide necessary energy to overcome these barriers. Regulation of enzyme. Maintain redox balance. Role of NADH. Reduction of oxaloacetate. It converts OAA to malate. Maintain redox balance. Enzyme regulation. Energy coupling.
We wrap up that gluconeogenesis is a vital metabolic pathway that allows the body to maintain blood glucose level during periods of fasting, low carbohydrate intake, or intense exercise. Thank you.